Welcome to Research Roundup, a quick dive into recent studies that connect directly to our testing and how they inform clinical practice.
So this review article summarizes that as much as 30% of patients with IBS with diarrhea are now recognized as having potentially bile acid malabsorption. Here's an example of a patient that actually had increased bile acids in the stool, and that's typically consistent with diarrhea or loose stools. You can see here the total bile acids are high for this patient, as well as the primary bile acid. So those are two indicators suggesting this patient may have faster transit. Certainly in patients that do have fast transit or diarrhea, this could be one of the factors that is contributing to their scenario. And then on GI-MAP, we often will see a pattern related to that in the commensal section. This is actually something that's been noted in research. Often you'll see patients that have certainly in IBS-D in general, and even in inflammatory bowel disease, there's a tendency towards increased E. coli, which is Escherichia. And at the same time, patients often will have deficient E. coli bacterium pneumoniae. Now, this is actually a recognized signature that many IBS-D patients and patients with inflammatory bowel disease tend to have these imbalances in their commensal. Interestingly, this has been linked potentially to mucosal biofilms that have been found in the ileum and the first part of the colon. So this is very interesting research. It was published recently. In this study, they found that almost 2/3 of patients with IBS and up to 1/3 of patients with ulcerative colitis had these endoscopically visible biofilms. So when they analyzed the biofilms, they found an increase in E. coli, which is Escherichia, and also an increase in primary bile acids and overall fecal bile acid excretion. So it may be that these biofilms and this overgrowth are related to potentially causing or somehow involved in this bile acid malabsorption phenomenon. And this is just an illustration depicting what they found that in patients, generally healthier patients that did not have biofilms, there's a good bacterial diversity and good levels of Faecalibacterium. But in the patients that had predominance of biofilms, they found a decrease in bacterial diversity, decrease in Faecalibacterium prosnitzi, and also this increase in E. coli and primary bile acids. To further underscore this association, especially in inflammatory bowel disease, this was a recent study published just a few months ago, where they linked an increase in bile acids in the colon to an increase in these inflammatory type bacteria, including E. coli. That's noted here in the title. And they also say Klebsiella pneumoniae and E. coli are frequently overrepresented in patients with IBD, particularly during active diseases. They also note that Proteus is also commonly a part of that picture. They go on to say we found increased abundance of these inflammatory type bacteria in the colonic mucosa of patients with Crohn's disease, with a concomitant decrease in the transporters that resorb bile acids in the ileum. So again, this seems to be linked to this bile acid malabsorption scenario, which in turn then seems to not only promote looser stools, but may also further promote inflammatory dysbiosis. So it may be part of the overall pathology in inflammatory bowel disease, at least for a subset of patients. Also, NAC, N-acetylcysteine, has been shown in several studies to also disrupt biofilms and may counteract various inflammatory microbes in multiple ways. It's noted here in this study that basically NAC helps to modulate LPS-induced intestinal dysfunction, basically also help improve the epithelial barrier function. So certainly there's some potential utility for NAC in some of these cases. Also cinnamon, so various plant extracts, but particularly cinnamon has been well studied as a key biofilm disruptor. And that's noted here in this review article, where they looked at specific mechanisms for cinnamon and other factors. And then lastly, here's another example that this title here is New Insights into the Antimicrobial Action of Cinnamaldehyde, which is an extract from cinnamon towards E. coli, and then its effects on intestinal colonization in this animal model. So There's actually lots of different ways to help counteract some of these inflammatory microbes and also possibly reduce the biofilms that they often produce that can be part of the pathological picture for some patients.